Hello, everyone. My name is Noelle Thundathil, and I will be presenting my summer project on a molecular genetic investigation into the origins of anthelmintic drug resistance in Ancylostoma caninum in pet dogs across the USA. So Ancylostoma caninum is the most important gastrointestinal parasitic nematode infecting dogs in the USA. Infection from hookworm can result in different pathological consequences like anemia and enteritis. Infections from A. caninum larvae can occur through percutaneous invasion or ingestion, and hookworm larvae, once they have infected their host, can mature and pass their eggs through the host feces into the soil to restart the cycle of infection. These hookworms are typically well controlled by anthelmintics, specifically benzimidazoles that are approved for use in canines. However, cases of recurring canine hookworm infections have increased by 47% between 2012 to 2018 in the USA, and recently, high levels of benzimidazole resistance in greyhounds have been reported. So it's been well established that the isotype 1 beta-tubulin locus is associated with benzimidazole resistance, and that the most commonly reported mutation associated with resistance is found at codon 167Y. So these figures show the high presence of the 167Y mutation in A. caninum infecting greyhounds and pet dogs, as seen by the frequency of the red bars. On the left is the greyhound plot, where the presence of the 167Y mutation is present at a much higher frequency when compared to the plot for the pet dogs, as seen on the right. This shows that there is higher resistance to drug treatment in greyhounds than pet dogs, and that resistance could have spread from the greyhound population to domestic pet dog populations. So we hypothesized that resistance could have originated in greyhounds and spread to the pet dog population. So it is a well-known fact that racing greyhounds have unrestricted access to dirt and sand tracks that compose the ideal environment for hookworm larvae to thrive. Since the dogs are exposed to this environment quite regularly, they are often subject to intense deworming protocols that would present a very high pressure for drug selection on the hookworm populations on these farms and racing kennels. Retired greyhounds from these breeding farms are often rehomed to different places in the US, which increases the chance for A. caninum environmental contamination and the spread of resistance to domestic pet dog populations. So the working model as of now is that drug resistance emerged in greyhounds due to their extensive treatment with anthelmintics and then later spread to the pet dog population. Now, if that is true, we would expect to see lower genetic diversity at the beta-tubulin locus in A. caninum in greyhounds than pet dogs, but see no such difference in neutral mitochondrial markers. So for this project, 2 neutral mitochondrial markers were sequenced that would allow us to compare the genetic diversity in greyhounds versus pet dogs. So we sequenced 120 pet dogs and 50 greyhound hookworm fecal samples from 4 different regions in the US, and designed primers to target different regions of the COX1 and NAD1 mitochondrial genes. Then, using the primer pairs we generated, we were able to produce PCR amplicons that were sequenced using the Illumina MiSeq platform, following which we used the DataTube bioinformatics pipeline to generate a table that would help us compare the genetic diversity between A. caninum in greyhounds versus pet dogs. So in this project, we were utilizing next-generation sequencing technologies and new data analytics pipelines that were not previously explored to generate our final results. So on the bottom, we can see the COX1 mitochondrial marker genetic diversity in both greyhound and pet dog populations. And on the top, we can see the beta-tubulin diversity for the greyhound and pet dog populations. These results show us that there is much higher diversity in the greyhound population in the mitochondrial markers compared to the greyhound population in the isotype 1 beta-tubulin marker, as seen by the presence of the colorful bars in the greyhound mitochondrial marker plot and the lack of colorful bars in the greyhound beta-tubulin plot. This is consistent with the hypothesis that selection originated in greyhounds and that there is stronger selection for resistance at the beta-tubulin marker in greyhounds than pet dogs. These results are useful for future anthelmintic treatment administered to pet dogs and have implications for treatment in humans as well. With that, I would like to thank Dr. Gilliard and the Gilliard Lab, as well as the Host Parasite Interactions Research Training Network for their ongoing support. Thank you.